The human brain. In adults, about 3 pounds of flesh holds a key to life. It controls our entire body and holds our memories and thoughts. But how did we end up discovering the true functions of such a delicate organ? The 1800s was a century full of revolutionary discoveries about the human brain. Before then, physicians and scientists didn't know the full extent of what the brain and its structures were responsible for. In fact, many discoveries resulted from physical accidents or unethical experiments. One example is Dr. Robert Bartholow's brain experiment done on Mary Rafferty. In 1872, Mary Rafferty was a regular woman from Cincinnati working as a servant in a household. She had decided to go to the Good Samaritan Hospital, where Bartholow was practicing as a doctor, to get a small ulcer or open wound checked out. The ulcer was located on the top of her skull and was over 2 inches in diameter. This opening was caused by constant friction from the whalebone in a wig she had worn for years after experiencing a scalp burn. Rafferty became an outpatient to monitor the ulcer. She was diagnosed with epithelioma, a form of cancer originating in epithelial cells which can be found in various tissues in the body. In 1874, Bartholow realized he had a clear view of the brain, and because the injury hadn't impacted any of Rafferty's senses, he decided to use an electrical current to stimulate different parts of her brain and meninges. Bartholow was very interested in the excitability of the cortex, and he wanted to learn new findings and confirm previous experiments. For example, at that point, electrical stimulation of the brain had been performed in other mammals, such as dogs. David Farrier, a neurologist who had stimulated the brains of dogs, had concluded, all parts of the brain in front of the fissure of sylvius, the anterior lobe, have to do with certain motor actions of the paws, legs, tail, and facial muscles, and the muscles of the tongue. The corpora striata are motor in function, and electrical excitation of them causes muscular movements of the opposite side of the body. The corpora quadrigemina are concerned in vision, and strangely preside over the extensor muscles. Therefore, Bartholow had an aim, to test the excitability of the cortex and compare what happens with 2 different electrical currents. Once Rafferty consented to the experiment, he used electrodes to first stimulate sensibility of the dura mater, one of the layers protecting the brain. Then he inserted the electrodes into the posterior lobes at different currents. Initial stimulation of both sides produced contractions of the limbs, turning of the head, tingling sensations, and a pain in her hand. After this, Bartholow increased the current, resulting in frothing of the mouth, convulsion, and a temporary coma. 2 days later, Rafferty was brought back for further testing, but was noted to be experiencing difficulty walking, a pale complexion, and blue lips. On day 6, Rafferty became unconscious, dying sometime later. Bartholow also performed an autopsy on Rafferty's brain, identifying the regions of the brain in which the needle penetrated. So besides ending the life of an innocent woman, what did Bartholow learn from his experiment? Today, Bartholow is considered one of the first individuals to have stimulated the human brain by using electrodes. He was able to demonstrate that the brain is able to be stimulated by direct electrodes and the localization of motor and sensory function. Scientists today have estimated that the electrodes inserted by Bartholow were inserted into the upper parietal lobe, which is mainly responsible for processing sensory information and language. However, neurons in the parietal lobe also hold axons belonging to the pyramidal tract, a branch of nerve fibers that is responsible for voluntary muscular movements. Although it is still debated what exact regions of the brain contribute to the observations in Rafferty, it has been concluded that the movements were due to a disturbance in the cortex almost exclusively. This means that the cerebrum and its various lobes, excluding known deeper structures, are responsible for movement. This experiment did not only influence the medical community, but demonstrated how a physical change in the brain can change how we think and behave, an important aspect of the science of psychology that was newly growing. Before, we didn't know where our thoughts or consciousness originated. We didn't know if the mind controlled the brain or vice versa. Therefore, this was a huge step in discovering that the brain was responsible for the development of not just our actions, but the decision to act and the ability to think. What ended up happening to Bartholow anyways? He must have been canceled, right? Even though he received a lot of backlash and published an, quote, apology, end quote, he ended up being a very successful and praised neurologist up until he died. This was even after the American Medical Association published their own statement apologizing for Bartholow and stating they do not condemn his actions. A common misconception is that he inspired an era of evil surgeons who experimented on human patients. Although Bartholow didn't inspire this era unfortunately began about 8 years later through neurosurgeon William W. Keene. One good thing that did come out of Bartholow's experiment was law reform concerning vivisection, or the act of performing live operations on animals for the purpose of science. In fact, in 1876, the UK passed a bill that restricted experiments on animals. Although scientists today have benefited in learning more about the brain from Bartholow's experiment, he has only ever concluded, quote, "It has seemed to me most desirable to present the facts as I observe them, without comment," end quote. Indeed, his experiment and the death of Mary Rafferty left many without comment. It makes one wonder whether our knowledge of the human brain and body today would be the same without the horrible mistakes from the past.